Okay, so imagine this. You have a cancer drug that's so potent it can literally starve a tumor. But there's a catch. It's so difficult for the body to actually absorb that its full power remains locked away. Well, today we're talking about how to unlock it. We're going to dive into how nanotechnology is completely re-engineering the way this anti-cancer agent, Fruquintinib, travels through the body, turning a huge biological challenge into a real therapeutic triumph. And this really gets to the core of it. We have this powerful weapon, Fruquintinib, but its potential is completely shackled by its own pharmacology. As you're about to see, nanotechnology offers the key to break those shackles. It's about changing the rules of the game. All right, here's our roadmap. First, we'll set the stage with the clinical problem that doctors are facing every day. Then we'll meet our molecular sniper, Fruquintinib, and really dig into its Achilles heel. After that, we'll introduce the elegant nano solution. Look at the proof, because the data is compelling. And finally, ask the big question. What does this all mean for the future of cancer treatment? So let's start with the clinical reality. We're zeroing in on metastatic colorectal cancer, MCRC, in a very specific, very tough situation. What happens when the disease just stops responding to the standard treatments? In these third and fourth line settings, oncologists are in a difficult spot. The tumors have become highly resistant. The therapies we have left might offer only moderate survival benefits, and often that comes at the cost of really significant dose-limiting toxicities. This creates a super narrow therapeutic window and a clear, urgent need to get more out of the powerful drugs we already have in our toolkit. And right into this challenging scenario steps our molecule of interest, Fruquintinib. And you can really think of it as a targeted therapy that acts with the precision of a molecular sniper. So what is it? Fruquintinib is a highly selective tyrosine kinase inhibitor of the vascular endothelial growth factor receptors, specifically VEGFR1, 2, and 3. Its mechanism is incredibly precise. It competitively blocks the ATP binding pocket on these receptors, which just shuts down the whole signaling cascade that drives tumor angiogenesis. Basically, it cuts off the tumor's blood supply, starving it of the nutrients it needs to grow. And look, this isn't just a theory on a whiteboard. Fruquintinib's efficacy has been proven, and proven robustly in major international clinical trials. You've probably heard of them, FRESCO and FRESCO-2. These trials established it as a legitimate weapon in the fight against refractory MCRC. So you have to ask, right? If we have this potent, clinically validated drug, what's holding it back? Well, the problem isn't its mechanism of action. The problem is its fundamental pharmacology, what we're calling its biological Achilles heel. It really boils down to three core barriers. First, it's highly hydrophobic. It has a high log P value, which just means it hates water. It doesn't dissolve well in the gut, so absorption is low and all over the place. Second, whatever little bit does get absorbed gets hammered by what's called first-pass metabolism in the liver, specifically by CYP3A4 enzymes. They just chew up the drug. And third, the drug that's left can go to the wrong places, causing systemic toxicities like hypertension and proteinuria. Put it all together, and its therapeutic window gets squeezed down to almost nothing. But this is exactly where some clever pharmacokinetic engineering comes in. So let's explore the nanomedical strategy here, self-emulsifying drug delivery systems, or SEDS. The best way to think of it is as an intelligent vehicle for a very, very difficult passenger. The whole principle is actually remarkably elegant. First, you take Fruquintinib and dissolve it into what's called a pre-concentrate. It's an optimized mix of oil, a surfactant, and a co-surfactant. Now, when this mixture hits the watery environment of your stomach, it spontaneously forms a stable nanoemulsion. We're talking tiny, drug-loaded droplets, usually under 200 nanometers. It effectively repackages that water-hating drug into a form the body can actually absorb. Now, here's the real magic. This is the game-changing part. A standard oral drug gets absorbed into the portal vein and goes straight to the liver to be metabolized. But this nanoemulsion gets absorbed differently. It hitches a ride on the intestinal lymphatic system, the body's natural highway for absorbing fats. These little nanodroplets get packaged into chylomicrons, travel through the lymph vessels, and enter the bloodstream directly, creating this beautiful bypass around the liver. That destructive first-pass metabolism completely avoided. Okay, that's a brilliant theoretical advantage, for sure. But the real question is, does it actually work in a living system? Let's take a look at the preclinical evidence. And really, it all comes down to one single number, 4.8. And this chart shows you exactly what that number means. In rat models, when they delivered Fruquintinib using this nano platform, it resulted in a 4.8-fold increase in total drug exposure, what we measure as the area under the curve, or AUC, compared to the regular free drug. That's not a small bump. That is a dramatic enhancement in bioavailability. And of course, that massive increase in bioavailability translates directly to what we really care about: superior anti-tumor efficacy. In a colorectal cancer xenograft model, you can see the nanoformulation achieved significant, sustained inhibition of tumor growth. It just completely outperformed the free drug at the same dose. And what's more, by improving the delivery and bypassing the liver, this whole approach also leads to a much better safety profile. We see reduced markers for both kidney and liver toxicity, lower creatinine and lower ALT and AST levels. And what does that mean for the patient? It suggests a much wider therapeutic window. It potentially allows us to use higher, more effective doses with less toxicity. That is a huge win. So really, what we're looking at is a completely new therapeutic equation. By solving the solubility problem, engineering that liver bypass, and enhancing tumor accumulation, we can fundamentally shift the entire efficacy to toxicity ratio. It's a new paradigm: maximum efficacy with minimum toxicity. But let's be realistic. The journey from these incredibly promising results at the bench to a real therapy at the bedside is, well, it's not easy. It's fraught with challenges, a path often called the translational valley of death. You know, to bring a complex nanomedicine like this to market, you have to navigate a whole series of critical hurdles. You have to prove you can manufacture it at scale under strict GMP standards. You have to guarantee its long-term stability. And you have to overcome some pretty significant cost and regulatory complexities before you even think about starting clinical trials. And all of this leaves us with a final, really provocative question. This isn't just about Fruquintinib anymore, is it? Could what we're looking at here be a true platform technology? One that's capable of unlocking the potential, or maybe even resurrecting, an entire class of powerful but pharmacologically challenging drugs? Because if the answer is yes, the impact on oncology could be absolutely profound.